research in affiliation with University of Otago. Before launching into his PhD, he studied a Master's in Plant Science at Glasgow University in Scotland. Without further ado, thank you.
Thank you, Kevin. Good afternoon, one and all. In this presentation, I will be discussing more about molecular mechanisms involved in mobility of mobile genetic elements or mobile genomic islands, known as horizontally acquired islands in the phytopathogen Vectobacterium heterosepticum. As mentioned by earlier speaker, Dr. Andrew, Vectobacterium heterosepticum is a gram-negative and enterobacterial phytopathogen. This is a seed board, and its virulence is pathogenicity is only restricted to potato plants. It has very narrow host range. This bacteria causes mainly tuber maceration known as soft rot and stem necrosis known as blackleg. Apart from these two diseases, this bacteria can also cause post-harvest lenticle rot. This is becoming more concerned in many temperate potato growing regions these days. Many of the virulence factors associated with this bacteria has been identified. Here, this picture showing the illustration of a Vectobacterium heterosepticum cell and its virulence factors. This bacteria produces plant cell wall degrading enzymes such as cellulases and pectinases. This also possesses all types of microbial protein secretion systems such as type 3. As previously described by previous speaker, Andy, this bacteria produces a very key important phytotoxin known as cornofrassic acid. Many of these virulence factors have been shown to be associated with mobile genetic elements. PBS strain SCRA1043 has many such mobile genetic elements which are thought to be involved in transferring pieces of DNA from one bacteria to another bacteria. Mobile genetic elements found in a chromosome are known as horizontally acquired islands, and these horizontally acquired islands can be identified using comparative genomics. Here, this wheel-like diagram showing genome sequence of a PBS strain SCRA1043 in the outer circle, and comparing sequences with genomes of enterobacterial animal pathogens, non-enterobacterial plant pathogens, and plant-associated bacteria. As mentioned earlier, with the comparative genomic analysis, we can identify these mobile genetic elements. For example, HAI2 here is only present in PBA SCRA1043, but absent in all other bacteria. This demonstrating that these particular islands are thought to be acquired via horizontal gene transfer and thus playing very great role in the evolution of this bacterium. HA2 shares many characteristic features with click family of horizontally acquired islands. These click family of horizontally acquired islands have been found in many pathogenic and non-pathogenic bacteria. Moreover, all click family of horizontally acquired islands shares many characteristic similar features, such as they all located at certain locations in a chromosome, like tRNA sites. These all family of horizontally acquired islands delineated by direct repeats at both the ends, which are likely to be involving in site-specific recombinations of DNA. And these all click family of horizontally acquired islands possess a very conserved integrase gene at either end of the chromosome, either end of the island. More oftenly, they contain lower GC content than the rest of the chromosome. During horizontal gene transfer, these mobile genetic elements or horizontally acquired islands shown to be excised from a chromosome. Upon accretion, they form a circular intermediate containing FP site. Upon accretion, chromosome will be repaired and tRNA site will be reconstituted, resulting in formation of FB upon accretion of this particular mobile genetic elements. Sorry. And this actual excision process can be identified by amplifying FP site, and similarly reconstructed tRNA or repaired chromosome can also be amplified by FB sites using PCR. This actually allowing us to monitor the excision process of HAI2 from its bacterial chromosome. Here this gel picture showing PCR amplified product of HB and HP, which is indicative of excision exiled form of HA2 and the reconstituted form of tRNA. Once we detected HA2 excision by using PCR, we wanted to quantify the frequency of HA2 excision from its population of cells. For that, we compare the formation of FB and HP using a single copy gene which is located in a chromosome, which is a ECA0515, is about 200 base pair. PCR amplified product is about 200 base pair in length. And the difference between amplification of FP and HP against a single copy gene is being expressed in terms of ratio. Here, this graph demonstrating formation of a ratio of HP and HB in a wild type SCRA or in a wild type PBA SCRA 1043 grown in medium. As we can see, the formation of ratio of HP is about 10 to the power of -5 to -6 copies of HP per cell. Therefore, in a population of about 10 to the power of 8 cells, about 10 to the power of 3 cells are undergoing this HA2 excision process at any given time. Similarly, as we can see, there are about 10 to the power of -4 to 10 to the power of -6 copies of HB per cell, which is indicative of repaired chromosome. Which means in a population of about 10 to the power of 8 cells, about 10 to the power of -4 to 10 to the power of 10 to the power of -2 to 10 to the power of -4 cells are undergoing this repaired process at any given time. Once we quantified HA2 frequency by growing wild type in media, we wanted to quantify HA2 excision frequency in its natural host, which is potatoes. For that, we have grown wild type bacteria in planta and in media, and we quantified the frequency of excision. Here, this graph showing ratio of HP and FB. As we can see, there are about 10 to the power of -2 to -1 copy of FB and HP formation when wild type grown in planta. But whereas this formation of FP and FB is about 10 to the power of -5 copies per cell when the same bacteria grown in wild type in medium. Once we quantify the HA2 frequency by growing wild type bacteria in media and in planta, we have been looking for the genes in island which are thought to be involved in this HA2 excision process. For that, we have studied number of genes. Today, I will be presenting the result of only one gene, which is conserved integrase gene. For that, we have silenced integrase gene, which is located within HA2 region. And we have grown wild type bacteria and integrase mutant in planta and quantified HA2 excision. Here, this graph showing ratio of HP and HB. As we can see, there are about 10 to the power of -2 to -1 copies of HP and FP per cell when wild type bacteria grown in plantum. But whereas formation of HB and FP is completely abolished when integrase mutant grown in plantum. This clearly demonstrating us that HA2 excision is mediated by integrase. Integrase is known to be mediate site-specific recombination between two DNA recognition sites such as FP and HP. From this, I conclude that HA2 can excise from bacterial chromosome. HA2 possesses tyrosine family integrase, which is likely to be involved in the recombination events that enable successful extraction of HA to implantum. We are continuing to study the genes in island which are involved in this, which might be involving in this HA to extraction process, which is playing a very great, incredible role in evolution of this bacterium. And I would like to acknowledge my supervisors and molecular bioprotection lab members at Plant and Food Research New Zealand, and the funding agencies supported this research. Thank you very much, and thanks for providing this opportunity to the organization. I would like to take any questions.
That was great. Worn out after lunch. I guess, how do you feel that with all these pathogens, the elements that are flying around in our bacteria now, how do we relate now to the whole species concept when we talk about plant pathogenic bacteria? I mean, what's the relevance today of some of our traditional taxonomy, given that the biology is perhaps so influenced by the pathogenicity of?
Okay, this is very rather global structure, because one question is, this is an enterobacterial bacteria in the beginning, but it became a phytopathogen. And to become a phytopathogen from an enterobacterial, these mobile genetic elements, HA2 and all horizontally acquired island, played a great role and causing such a devastating diseases across the world. I mean, this kind of correlation, and from this is happening, many of pathogenic bacteria across, could be in humans and plants and animals. And
no, thanks for that. Thank you once again.